Okay, welcome everyone to the PhD seminar from Monica Olson. I want to tell you a little bit about a background about Monica. There's a few pictures of her here showing her processing some of the corn several years ago. And I really want to emphasize that Monica has completed now a 7-year journey to finish her PhD entirely part-time, because she has been working full-time at what was originally Dow AgroSciences and then was the Corteva AgroScience in the last few years. She's been based in the Indianapolis office or campus, you might say, and she started in the Indianapolis office of Dow AgroSciences in 1999. She had graduated from the Indiana University in Bloomington, and she's in fact in the biology area primarily. And she's in fact the first graduate of Indiana University that I've allowed into Purdue. So we hope that that ends well, because it certainly began very well. And so she started in January of 2014, directly from the bachelor's degree and into the PhD. And although it was a long haul, it was definitely one that had some very exciting moments. And so I'm thankful that all of you are able to hear some of the highlights today. But I would be neglect if I didn't say that the first word in her thesis title is hyperspectral, and there's no way that that thesis would have been able to begin with that title were it not for the considerable assistance that we've received over the years from Dr. Melba Crawford. So having said that, Monica, I'm going to stop sharing and I'm going to ask you to start your seminar, please. Thank you.
Okay, great. So I will go ahead and stop my video so I'm not distracting myself, but I will share the screen. And Tony, it might help if you can mute everybody.
As long as that doesn't
except for me.
Right. Let me just see whether or not that's a possibility here, and then we'll go from there.
And then while you're doing that, hopefully my screen is on presentation mode with a single screen.
You are in good shape that way. You are now in presentation mode. So just proceed. I think you'll be fine.
Great. Well, thank you so much to everybody for coming to my presentation. I really appreciate you spending this time here. So I will be talking about hyperspectral imaging for estimating nitrogen use efficiency in maize hybrids. So nitrogen is an essential nutrient that's needed for growth in agriculture. And if you've been driving around Indiana, I'm sure you've seen a lot of corn fields. And if you peered in between the rows near late in the season, you would still see nice, strong, stiff stems, big leaves, and a really nice full ears. However, if this was a field with insufficient nitrogen, instead you would see a lot of dead leaves, thin stems, and a lot of barren plants. So even though this essential nutrient is needed for growth, it really adds to farmers' costs. And there's also potential environmental problems due to misapplication or overapplication, or issues such as leaching into the water of nearby fields. Using more nitrogen efficient maize hybrids can result in lower nitrogen fertilization applications, as well as increasing yields in poor environments with less input. Developing nitrogen use efficient hybrids, however, is really costly and laborious because of all the time it takes of sampling in the field, as well as processing all the samples in the laboratory. So we really need to improve agronomic and plant breeding methods in order to assist in selections for superior NUE hybrids. So I've talked about NUE. What do I mean by NUE, or nitrogen use efficiency? Well, these are measures or parameters that have been developed to study and communicate about nitrogen translocation and utilization in the plant. Specifically, I will be talking about NIE, or nitrogen internal efficiency, which is the amount of granule produced per unit of nitrogen in the plant. I will also talk about nitrogen conversion efficiency, or NCE, which is the amount of biomass produced by the plant per unit of plant nitrogen. Now, it's important to note that later in the season, NCE, in regards to the biomass, includes the above ground plant material as well as any grain. To give you some historical context, Capably and Vine and Mueller and Vine have studied old era hybrids and new era hybrids. The old era hybrids were those produced prior to 1990, versus the new era hybrids were those produced after 1990. And even though these are being grown at the average same nitrogen fertilizer rate, they're actually being planted at higher densities, which is one of the reasons that yields have gone up for the newer hybrids. Comparing the NIE values for the old versus the new, is these new era hybrids have also increased in efficiency. One of the ways that the plants have compensated for that is that they've actually put less nitrogen in the grain. So for this seminar, we had to focus on our objective, which was to evaluate in-season methods for predicting end-season nitrogen efficiency parameters. What in-season methods did we look at? We looked at traditional methods and modern methods. For the traditional methods, we looked at field-based physiological measurements, while for the modern methods, we looked at hyperspectral imaging. With both of these, we wanted to predict these nitrogen efficiency parameters. For the physiological measurements, we focused on NIE. And for the imaging, we focused on nitrogen concentration, nitrogen conversion efficiency, and the NIE and the nitrogen internal efficiency. So let's focus on the field-based physiological measurements effort that we looked at. Our hypothesis was, because this in-season physiological measurements are secondary traits which describe the growing status of the maize plant, they are correlated to and can serve as surrogate measurements of the nitrogen status with a specific focus on the NIE. So what physiological measurements did we capture? At R1, when the plant was flowering, we obtained plant height, stalk diameter, and then used a Konica Minolta handheld meter to measure the SPAD of the ear leaf. At R2, R3, we took 2 green leaf counts and another ear leaf SPAD measurement. And at R5, we used a LICOR plant canopy analyzer to measure the leaf area index, which is the amount of plant leaf area relative to the ground surface area. In order to obtain our nitrogen parameters, our NIE, we collected biomass at R6. We took 10 plants from the center 2 rows, weighed everything, ground everything, and then sent everything to the laboratory for Demas combustion in order to determine the nitrogen concentration and thus determine the nitrogen content. So this experiment started in 2015, and we planted fields in Acre in West Lafayette, as well as at PPAC up in northern Indiana. We had 285 hybrids, which all had been crossed to a common female tester. The germplasm was 3 sources: USDA, checks, and Dow AgroSciences. And everything was either early maturity group or a late maturity group. Everything was planted in triplicate, except for the checks, which we actually had 6 to 10 of those. We had 4-row plots, and they were about 8 meters long. And our planting density was 89,000 plants per hectare. We had everything in a modified augmented design, which means that those checks were planted throughout the field multiple times. And because we had so many hybrids, we only had one nitrogen rate, 67 kilograms of nitrogen per hectare, which was applied as urea ammonium nitrate at pre-plant. So what were our results? Here on the left is a histogram of the NIE response that we saw. On the x-axis, I've broken it up by location, acre, and PPAC. And on the y-axis, we have the two maturity groups, early and late. So what you notice is that overall, the mean NIE values at acre were higher than at PPAC. But in general, we had a very large NIE response from around 50 to around 160. And remember, this was under only one nitrogen rate. On the right, we have the same histogram, but instead we've broken it up by source. And what you'll see overall is that the mean checks had higher NIE than the agrosciences germplasm, which had higher NIE values than the USDA. So our NIE response varied by germplasm. So now we wanted to use these phenotypes in order to predict NIE. On the x-axis here are the various phenotypes that we looked at, and on the y-axis, you will see the Pearson correlation coefficient that we calculated. In general, what you'll first notice is that all the physiological measurements that we took were negatively correlated to NIE. Plant height was moderately correlated to NIE, but there were no physiological measurements that were strongly correlated to NIE. So that was our effort at looking at the physiological measurements. So now let's look at the modern methods, or that hyperspectral imaging. What is hyperspectral imaging? It is reflectance data that's collected in a wide spectral range from around 300 to around 2,500 nanometers in very narrow bands, which can range between 1 nanometer up to 70 nanometers per band. And this has been well studied for sensing both biochemical and cellular components, as well as leaf characteristics such as leaf thickness, or even canopy characteristics such as foliage density. There are pros and cons to hyperspectral imaging. Because we have such high spectral resolution, we can really detect subtle differences between materials. Also, because we're acquiring the entire spectrum, it means that we need no prior knowledge of the materials that we're studying. Since it's imaging, it's obviously non-destructive. And then there's a lot known about the differentiation in the spectral signature between vegetation and soil. What do I mean by spectral signature? If you look at the graph on the top right, the x-axis has the wavelength and the y-axis has reflectance. And what you see is that soil has a relatively flat and low response for reflectance across the spectrum, whereas vegetation has a peak around the green portion of the spectrum, a dip in the red region, and then high levels of reflectance in this NIR, or the near infrared region. Unfortunately, hyperspectral imaging equipment is very costly. And also, there's a lot of multicollinearity in the data, which means that both the neighboring pixels and the neighboring spectra are related. What this means is that it really exponentially increases the number of training pixels you need in order to have confidence in the model, which means more sampling out in the field. Because you've got so many wavelengths, you've got very large data sets, and then this multicollinearity that I talked about can result in a complex data analysis. So our hypothesis was that because hyperspectral imaging is an integrated view of the plant, in-season hyperspectral imaging can predict the nitrogen status of maize hybrids with a focus on NIE and NCE. So because of the problem or the complexity of the data for the multicollinearity problem that I talked about, we dealt with this in two approaches. First, we looked at hyperspectral indices, and then we looked at partial least squares regression. But first, let's talk about the materials and methods that are specific to this imaging experiment. The experiment started in 2014 in California, where 9 mycogen hybrids were planted in 2 fields near Sacramento, California. Everything was irrigated and maintained as well watered and grown under 3 nitrogen rates and 2 planting densities. We had 3 to 4 replicates, and everything was planted in a randomized complete block design. For the imaging, this data was obtained from a manned aircraft. It had 50 centimeter spatial resolution and around 5 nanometer spectral resolution. The plots were 6 rows wide and 12 meters long, and phenotyping for both biomass and nitrogen content was collected at V12, R1, R2, and R6, and the imaging for from the manned aircraft was collected at V18 and R1, R2. This experiment continued in 2017 in Indiana when we planted 5 mycogen hybrids, which were a subset of that original 9. We planted everything in West Lafayette at acre, and everything was maintained as rain-fed. We used the same nitrogen rates and the same planting densities, had 3 replicates, and everything was a randomized complete block design. For the imaging, it was collected using a drone. It had 4 centimeter spatial resolution, which we downsampled to 50 centimeters to match the California data. And then we had 2.5 nanometer spectral resolution, which we binned up to 5 nanometers to match the California data. The plots were 4 rows wide and 15 meters long. We did phenotyping at the same times as California, and imaging was collected multiple times throughout the season. But in this presentation, I'll be focusing on the data which match California, the V16, 17, and the R1. Some more details on the remote sensing. Both years used push broom sensors, and all data was corrected geometrically and radiometrically, and we converted to reflectance using radiative transfer models. Okay, so let's focus on the hyperspectral indices. Hyperspectral indices are basically mathematical calculations based on the reflectance as specific bands. For example, let's look at NDVI, Normalized Difference Vegetation Index, which is a common index you'll see in the literature. If you take the reflectance at 800 nanometers minus the reflectance at 670, and then normalize it by the sum of the reflectance at those bands, you have your index. So what we did is we looked in the literature to see what indices have been studied. And we were interested in choosing indices that had been studied for both biomass, nitrogen, some type of grain yield or structural component, or plant stress. We chose 20 of these to look at for our study. So what do these indices look like? This is NDVI early in the season at V5 from our 2017 data. If you zoom in, what you'll see is that the plants are yellow and green and the soil is red. But remember, we're interested in only the plants. So for that, we needed to remove the soil pixels, which we use through thresholding. Once we had the plant pixels, then we calculated our indices by pixels and then averaged within each subplot. Okay, so let's talk about these nitrogen parameters, and I've just placed the calculations here for NIE and NCE as reminders again. Again, we had to do field sampling. We collected biomass of the 10 plants, measured nitrogen concentration based on mass combustion, and calculated nitrogen content. For comparison of our HSI models, we first needed to establish a ground reference model. For that, we used mixed models. For the fixed effects, we used the traditional agronomic factors of hybrid, nitrogen, plant density and the two-way interactions. And we use location and its two-way interactions as the random effects, with, of course, our Y variables being our nitrogen parameters. For the HSI models, then, we again use mixed models, but this time use the index, hybrid, the two-way interaction between those as the fixed effects, and then use location as a random effect, with the Y variables as for our N parameters. In order to select and sort through the 20 indices we had chosen for each image, we needed and established a decision tree. So first what we did is we looked at the correlation between our index and our nitrogen parameter at each of our locations, 3 locations. So if they were similarly correlated in directionality, that means they were mostly positively or negatively correlated, then we looked at the strength of the correlation. If they were moderately or strongly correlated amongst all 3, then we looked deeper into those. Any indices that didn't fit these criteria, we put them in the no further investigation bucket. Once we had that, those chosen indices, then we established those HSI models that I just talked about a bit ago. We made sure that these models fit the model assumptions of normality and constant variance. If they did, then we kept digging into them. Then we established whether we had, looked to see whether we had a significant hybrid effect. So our ground reference models are showing that we had significant differences between our hybrids for our N parameters. We're trying to use these HSI models to predict and differentiate between our hybrids. Thus, our models, our HSI models, needed to be able to show differences. So we dug into those. Once we had those, we looked at the statistics for each of the models and looked at a variety of statistics, such as ICAC's information criteria, residual values, and then looked for the best ones. Those, then we looked and moved to the hybrid ranking comparison. So again, we're trying to differentiate between our hybrids, and we looked at how did our HSI model rank our hybrids relative to our ground reference. Those that did a good job and comparable to the ground reference, then we started looking and did a visual inspection of the data of those predicted values. We were looking for models that had the smallest variance, and then those were the ones that we termed as our best HSI models. What did the data look like? What were our results? So shown here are the results for the best HSI models for predicting nitrogen concentration. First is our ground reference agronomic model. On the y-axis is the amount of measured nitrogen concentration. On the x-axis is the predicted nitrogen concentration based on the traditional ground reference model. And the black line is the one-to-one line. And you can see that this mixed model had really good predictions because there is very little scatter around the one-to-one line. On the right were the 2 best HSI models. You can see that around 1% nitrogen, we had pretty good predictions, but then we got some wide scatter at other points, meaning that the model was either over or under predicting the data. HREI 16 was actually a little better and had a little narrower scatter around the one-to-one line. So how did our hybrid rankings do? Well, first, let me talk to you. I am using constant format for this, so let me explain the format that I'll be showing. First, I have the hybrid name in a colored box, and to the right of that will be the Tukey means comparison level letter. And on the left of that, for the top hybrid or the best hybrid and the lowest hybrid, I will show the least square means estimate in units of whatever nitrogen parameter I'm talking about. In this case, it would be percent nitrogen. And for clarity, this is an estimate across 3 site years, 2 plant densities, and 3 nitrogen rates. Okay, so let's look at the results. For the ground reference model selected -6 and 9 as the top hybrids, and it differentiated 3, 4, and 7 as the lowest 3 hybrids. And as a reminder, we had a total of 9 hybrids. How did the HSI models do? Well, HBSI2 was able to identify -9, but missed 6, and it only identified -4 as a poor performer. HREI16 actually did a little bit better, identifying the 2 best hybrids, 6 and 9. It also found -3 and 4, but missed -7 and threw in -5. For NCE, we had the same format, the ground reference model again, and the HSIs on the right. And what you can see is at low NCE values, we had pretty good predictions, but as the NCE values went up, we had under predictions for our HSI models. And this is HBSI1 and HBSI2, and both of these had similar scatter around the 1-to-1 line. Looking at the hybrid rankings, -7 and 4 rose to the top for that ground reference model, and 1, 9, and 6 were at the bottom. For the HSI models, both of these gave similar results in the fact that they both identified -4 as a top performing hybrid, and were both able to identify -9 and 6 as low performing hybrids, but -7 and 1 were missed. For predicting NIE, we actually had 3 indices that were predictive. Again, on the top left is that ground reference agronomic model. And here to the right and bottom, we see the results for the HSIs. This actually is HBCI8 at the early time point, which gave good predictions, as well as at R1. We have another index, HBCI9, which performed well at R1, but not at V16. If you look at the scatter, these are actually pretty good, even at the higher NIE levels. So for these HSI models for predicting NIE, we're comparable to the agronomic ground reference model. How did our hybrid rankings look? These are the hybrid rankings for NIE, where 3 and 1 rose to the top, and 6, 5, and 7 were the worst. And overall, all 3 HSI models were able to pull out and differentiate between the top ranking hybrids and the low performing hybrids. So this HSI model for NIE was really comparable for the ground reference model at both the plot level and the hybrid rankings. In conclusion, our hybrid prediction accuracy was best for NIE, and we found 2 indices, a structural index and a plant stress index, that were pretty decent at predicting nitrogen concentration. 2 structural indices were predictive of NCE, and if you notice, both of those are focused on the near infrared to red bands. And then for NIE, where we had that best performance, these were 2 biochemical indices that are focused around the green to green portion of the spectrum, or green to blue. So that was our discussion around our effort for hyperspectral indices. Now let's talk about partial least squares regression. For the partial least squares regression, we needed to process the remote sensing data a little bit different. To extract our features, we still use the 5 nanometer bands, but we narrowed our cube a little bit and only used the bands from 410 to 920 nanometers. Instead of that thresholding technique that we used to remove the soil pixels, this time we used supervised classification based on support vector machines. Once we had our plant pixels, then we averaged our reflectance values to the plot level to obtain our spectral signature. Now we chose partial least squares regression because it allows analysis of such multi-collinear data without breaking any assumptions of the model. Because it's a machine learning technique, we split the data into train and test in a 70-30 split. And then we selected our factor loadings based on a 10-fold cross-validation. We looked at 12 models, 6 for each time point, and we looked at both full models, which included all 89 bands, as well as reduced models, which had fewer than 89 bands. Here's the data, and so let me walk you through it since you guys missed it, and I was talking to myself. So on the y-axis is a measured nitrogen concentration. Again, NCE for the middle charts and NIE on the bottom charts. For the x-axis is the predicted nitrogen concentration from the V16 time point, that earlier time point, and then the predicted value at R1. And the same thing for NCE predicted values and NIE predicted values. For the R-square values, what you want to do is focus on the R-square value, the second value here. And I forgot to explain that this is the one-to-one line. Okay? And if you look at the R-square values for all 4 of these plots, you'll see that all had R-square values greater than 0.65, and the coefficient of variance for these 4 plots was around 10 to 12%. If you look at the scatter around the one-to-one line, you can see that they're pretty consistent and they follow the one-to-one line pretty well. You will note that this graph here, this 4th graph for NCE, there's a few outliers. There's a few outliers here for the NCE. Once we looked into this, what we believe this is due to the transformation that we needed to do to the data in order to linearize it. So now let's look at the NIE values or the NIE models. And what you notice first, not only is there separation between the blue California data and the red Indiana data, but you also see that the models tended to predict everything the same. So regardless of whether you were dealing with an NIE value of 30 or 60 in California, it predicted it to be around 45, and similarly for Indiana. We dug into this and looked, and what we noticed is that both the red edge and the NIR bands were very important to the NIE model. And digging into those, we noticed that there was a clear separation or divergence in the reflectance values between California and Indiana. We looked at the residuals in those bands and noted that the assumption of constant variance at those bands was violated. So the NIE models were poor. What did our hybrid rankings from the partial least-squares regression models look like? So you've already seen the hybrid rankings for nitrogen concentration based on ground reference. Shown here is the R1 full PLSR model, hybrid rankings. So it was able to differentiate and pull out the top 2 hybrids. And then it identified DAS4 and 7 as lower performers, but missed DAS3 and instead threw in DAS5. For the nitrogen NCE hybrid rankings, you've already seen the ground reference rankings. So let's look at the R1 full PLSR for NCE. So this model was able to identify DAS7 and 4 as the top performing NCE hybrids. It also identified the lowest performing hybrids, 1, 9, and 6, but threw in DAS3 and 8 into the pile. If you use these models in order to identify those poor performing hybrids and remove them from the batch and see if you still have your good hybrids, what we found is that in eliminating the worst 3 hybrids, still left behind that best 2 hybrids were still in that pool that remained. So our recommendations for an early stage breeding program is to apply this imaging using either HSIs or PLSR methods to assist with hybrid selection. We believe that the imaging data provides information for decision-based selection, and we can use it to first find those low-performing hybrids and cull them from the list. Thus, at the next stage, you're testing fewer hybrids and increasing your efficiency of your breeding program. Next, we also recommend developing a multispectral camera with narrow bands found at those wavelengths that were useful for predicting NCE and NIE from our HSIs. The benefits to this is that not only is it easier and faster to process the data because you've got fewer bands, but also you can use cheaper equipment that you can have at multiple field stations throughout the world. So in summary, the hyperspectral imaging was predictive of the nitrogen parameters, but the feature extraction method used really affected which parameters could be predicted accurately. We recommend further validation and investigation into additional genotypes because some data suggested that certain genotypes may be less well predicted, but I did not show that data here due to time. We also suggest looking into additional environments. The data suggested that multi-locations are very important in nitrogen research. We were able to look at both Indiana and California, but additional environments is useful. And also additional imaging time points, especially later in the season at R3 and R5 would be useful to investigate since we were focused on that V16 and R1 time points. I'd like to thank all the students, both undergraduate and graduate, that have helped me in my time at Purdue, not only in sampling of the processing and sampling, collecting samples in the field, but also just answering my questions and really helping me out. Big thank you to Dr. Crawford's remote sensing team. Her students were great at trying to explain to me how to process this remote sensing hyperspectral data. Thank you to all the Purdue professors that answered all my questions. And obviously, great big thanks to my committee who have really guided me through my time at Purdue. Thanks to Dr. Boomsma for establishing those California trials, and to the Heritage Agri-Sciences people who did all the field station support in California. Thanks to all my Corteva colleagues who have really supported me through my journey. And lastly, a big shout out to Alyssa Batula, who was instrumental in teaching me how to do Python programming. So here are my references for the HSI indices, and now there's some time for questions.
Okay, so in this question period, feel free to use the chat or unmute yourself and pipe in orally if you wish.
Tony, are you going to be reading the chats, or do you want me to read it out?
I'll read the chats as they come in. I don't see any yet, except for the challenge we had earlier with the loss of audio for a little while. So.
Can I ask a question?
Of course.
This is Brock, Dr. Crawford, student. I noticed that, Monica, that was a very good job. Thank you so much for your presentation. My question is, I noticed you collect your leaf area index with plant canopy analyzer. I'm wondering if there is a reason you did that, or it was randomly to choose that specific instrument.
That is the traditional instrument we use in Dr. Vine's lab.
Okay, have you ever tried the other method, or the result would be the same with the other method? Have you ever investigate that as well or not?
Yeah, I know in your research you looked at LAI and looking at it through imaging. So if you're asking if we tried to use our imaging for LAI detection, we did not.
No, not imaging, actually. Another instrument is of like other instruments for getting LAI.
Nope, nope. The only equipment we used was the Lycor.
Okay, thank you so much.
You're welcome.
Is there another comment or question here? I don't see anything in the chat just yet. So maybe I could start. I mean, we're going to, as an advisory committee, have a 2-hour oral exam after this, and I don't really want to start with my questions for that period, but I can. So, okay, here's a question from Matt Bechdahl. Curious about temporal predictors. How early could you reliably find the relationships? So temporal predictors, and so here I suppose you could choose to answer that for, you know, any one of the nitrogen indices you talked about. And I think the other comment I'd like to make, just to add on to this as well, is I want you to say just a few words about the combination of high NIE and high yield in some of the top NIE hybrids, and whether the two ever came together or whether they weren't necessarily reflecting the same trend. So let's start first with temporal predictors. How early?
Yep. So obviously, you know, our focus was on that V16 R1 R2 time point, since that was the data that we had across both California and Indiana. Ideally, we would have been able to you know, have a broader data set. But looking at those two time points, what we saw is that usually the better models for NCE and NIE were at R1, so that later time point. And even visually, you could certainly see the change in the, you know, just looking at an RGB image, you could see a change in the response of the plants, just like you would see by eye, but you could certainly see that. And what was interesting is, you know, the sensing at V16 and R1, we usually see something visually later in the time point, depending obviously how poor how little nitrogen you're dealing with. So it was nice to actually even see it at R1. And then Dr. Vine, you talked about what was the grain yield level for the hybrids of interest. So what we actually noticed is that in general, the best NIE hybrids had high grain yield and either high or moderate nitrogen concentration. And then for the best NCE, usually tended to have lower nitrogen concentrations at R6.
And so what does that mean in terms of being able to predict something that has high NIE as well as high yield? Where would you go?
Well, obviously, you know, one of the fears when you're trying to predict, or when you're trying to go towards best NIE, is are you going to end up selecting low performing hybrids, even though NIE is the ratio of grain yield per plant nitrogen? You know, are you going to end up selecting for low yield, which is at the end of the line most important for the farmers? And this is actually encouraging, because I kind of thought, when I was just thinking about the question, I thought that the best NIE hybrids would have maybe moderate yield, but a few of the best hybrids did have that high grain yield. So that was interesting.
There's a new question here from Deanna, and she asks, have you thought about any other parameter that could help to improve the model? And I don't know, when this question from Deanna, whether she means the PLSR or whether she means something else, but I'll let you take it away. And I was also wondering if you could end your share screening, then maybe we would see you.
Okay, I can turn on the video, because yes. I turned it off so I wouldn't be distracted.
Right.
Let me.
We want to see how much you're sweating with these questions.
Okay, let me find my mouse first. Stop share. And then start video.
Perhaps it would just personalize it a little bit as we finish up here. Okay, so
yep.
So did you get the question from Deanna?
Yes, Yes, I see that. And we kind of, one of the thoughts we had was, especially for the partial least squares regression, since there seemed to be some separation by locations, we were thinking it might be useful to throw in some type of temperature or GDD or something to try and kind of represent the location, temperature, the growing conditions, really. So we were thinking that could be useful.
Thank you, Monica.
You're welcome.
Thank you for the question, Diana. Are there any other questions here? Maybe I'll go a little bit further then. So basically, you selected hybrids, averaged over densities, averaged over nitrogen rates. And if you were to adopt these conclusions in early stage breeding programs, would you automatically start with multiple N rates or multiple densities in order to do the most efficient job of selecting hybrids with higher nitrogen efficiency?
Yeah, so I think it kind of depends on which time point you are in your method. So if you are still kind of in the early stages of developing the method like we talked about, such as now, if you were, in a proof of concept and really trying to ensure that your predictions are accurate, then I would keep the multiple rates, at least a low, medium, high, or a low and a high, to try and ensure that your model is well stabilized across your spectrum of response. Ideally, eventually, you can get to the point where you're only using one N rate. And I would, you get better expression for your NIE, as we saw with the 2015 data, when you're testing at a low N rate. There we had obviously there was a lot of sources in that, but we had a great response there. But at the end of the line, you want a hybrid that can still produce under these low nitrogen conditions, but can achieve high yield levels when it is provided with the nitrogen concentration at appropriate levels. So initially, maybe while you're sorting through them, if you have a lot of hybrids, like at an early stage breeding program where you're really trying to sort through things, I would test at one rate. But I think eventually when you're really trying to characterize that hybrid, you need to evaluate it under at least a low and a high.
Great. Are there other questions from the audience here?
Hey, Monica, this is Mitch calling or speaking. I have just a general question for you. So one of the challenges that oftentimes creeps up with this type of research is that, especially when you have locations scattered across the countryside, that different farms and facilities have different platforms and cameras and protocols and ideas. How important is it that everybody have the same platform and the same camera? When I'm sure that as the results of your work become widely known, people are going to want to replicate that. How important is it that everybody uses the same type of instruments to collect these data? And if it's not that important, what would be what kind of recommendations would you provide to help the industry think about how to collect these types of multi-environment data sets?
I understand your question. And I think that was actually one of the questions, one of the challenges I thought with this data set was the fact that, the 2014 data was collected from a plane with hyperspectral camera, but a high quality hyperspectral camera, but obviously it wasn't the drone that we were using in 2017. And I was actually afraid that that would completely throw off any kind of potential analysis when I pooled the data together. So I was pleasantly surprised to actually be able to develop models that were able to use the reflectance data from both platforms, and honestly, different processing techniques and everything. So I think that's one of the values of the data analysis that we are providing. But you're right that, what equipment you're using is very important. And so I think first, you want to try and really focus on high quality hyperspectral images. So there's been work with looking at cameras, etc. But if you're really trying to compare, I think you're going to want to look at hyperspectral image versus maybe a lower quality equipment. I think those are all questions that need to be evaluated. I would, like the next step on this, I would still stay with some type of high quality hyperspectral image, maybe like that multispectral camera that we talked about, to try and allow, compensate for that cost that we're talking about. But one of the things that I noticed in processing the hyperspectral data is that I noticed how important it was and what a huge impact it had in how you process the data. What technique do you use to remove your soil pixels? How do you do you group your soil pixels first? Group all your plant pixels by plot, and then you calculate your index, or do you calculate your index for every pixel? So there's a lot of complexity and different things that can affect your analysis results at the end of the line. And so I think one is being really transparent about what processes you're following. But honestly, there's a lot of learnings that have happened. I remember a comment by Dr. Crawford that she said even in 2017, when her team was helping me collect all this data, even then they've learned things relative to what they're doing now. And so it's a really fast advancing portion of the field. And so it's exciting, but it can make it difficult to try, like you said, replicate results.
Nice presentation. Good answer. Thanks.
Okay, given the time and given the fact that you have other things to do, and we'd like to start our oral exam in a few minutes before Monica runs out of energy for the day, I'm going to say thank you all for listening into this presentation. Would also say that we've recorded this presentation and it will be available through the Vine Cropping Systems lab website next week, hopefully. And so there'll be additional opportunities to follow up with Monica in the weeks to come, and as she continues to work on her publications from this thesis. Thank you all for your attendance, and thank you, Monica, for a very well-structured seminar.
Thank you to everybody for attending.
All right, wonderful. I'm going to sign off here.